Shimokawa from university. So I'm from Tokyo, and I'm enjoying a beautiful summer here in Hokkaido. So today I'd like to talk about the application of knot theory in material science, but basically I will give you some very short introduction of knot theory, and we'll talk about application to knot theory to interlocking molecule. Here we will discuss supermolecule structure, which is made by links. And also, we discussed the topological change of the knot links in DNA and also vertex knots. So this is a picture of the interlocked molecule, which I will explain this in detail later. So we will discuss this. And also after that, we will discuss the topological change of the DNA knot links and also knot links in the vortex. So these are the contents of my talk. Okay, so first we'll start introducing the knot theory. So I'm a topologist and a mathematician. As a topologist, we mean that the knot is an embedding of the S1 that is just a circle into three-dimensional space. And if you put it in this way, then this is also knot, and this is called a trivial knot. And you can put it in a very nice way, such as this one and this one. This one is called turtle knot and figure eight knot, and so on. And if you have a multiple component, as in this figure, so in this case, you have two links. Those are called two-component links. And in biology or chemistry, sometimes it's called catenan, and this is called two catenan and three catenan and four catenan. So this is a three-component link and a four-component link. And I'd like to show you some nice picture of knots and links. So this is a software called Knotplot. And so this is a list of knots and links. So this is an example of the knot, and that is called 7-6. And when you consider a knot, we often draw a diagram as in this picture. And the important thing is that the number of the crossings in this picture. So here you see 1, 2, 3, 4, 5, 6, 7. And this knot is called 7-6 because it has 7 crossings. And 6 is just the index of there are 7 crossing knot, and this is 6 of those. And so these are examples of the two-component links. And so this is an example of the three-component link. And we will consider the topology of the knots and links. And this is not a good one. So sometimes you have a very complex knot, as in this picture, but as a topology, we will allow to move in a continuous way. That means that you can move a knot without intersecting or cutting. And if you can change using that move, we see that we say that those are the same knot. So in this picture, you see that this knot is changed into a trivial knot. So we will consider those knots are the same. So, and this one is similar to the previous picture, but if you change it, if you move it, it's a trivial knot. So this will give you some idea that knot theory is not so, knot theory is very easy to start, but if you'd like to do some research, sometimes it's very difficult. Even if you have a complete picture, it is not easy to check what the knot type is. This is one example. So this is called a perk knot. And for many years, knot theorists thought that those two knots are different, but it turned out that actually those are same. So this is the knot theory. So if you use software like this is called Knotplot, you can have a good idea what the knot theory is. And okay. So today I'd like to talk about the application of knot theory. And we will use some invariant of the knot. And I'd like to introduce just two of those. First one is called a linking number. So you consider a two-component oriented link. So here you see example of the two-component oriented link. Here we mean the oriented. That means that you have orientation of each component that is indicated by arrow. For this type of the link, you can define a linking number as in this picture. For each crossing, you put plus or minus 1 and consider the sum of those and take half of this. And this is called a linking number. And in this case, you have plus 2 in this picture. Here, you only consider the crossing between K1 and K2. And you can use this to check the equivalence class of knot links. And sometimes you can use it as a to say the chirality of the knots or not chirality of the links. And also, we'll often use something called Jones polynomial. Jones polynomial is defined also for oriented link, and this is the axiom of the definition of the oriented the Jones polynomial of the knots and links. The first axiom is that if you have a trivial knot, then the Jones polynomial is 1. And if you have 3 links that appear in this picture, so those are those L plus and L minus L0 are almost same, but only differ just one crossings. L plus has this, and L minus has this, and L0 has this. Then those 3 links, Jones polynomial of these 3 links have this relation. And using this, you can define a Jones polynomial. And that is very powerful tool, and we can use it for the determination of the chirality, for example. And if you have trifold knot, you have this, and the figurative knot, you have this. And Jones polynomial can be calculated using the axiom. So this is L minus and L plus and L0, and that will satisfy that axiom. And using those relation, we can calculate the Jones polynomial. But we don't go into the detail, but if you are interested in, you can see it in a notebook. Okay, so those are the very short introduction of the knot theory. And today I'd like to discuss the application of the knot theory to some real problem. The first one is considering the topology of interlocking molecule. So here, we like to consider the topology as in this picture. So these are made of the self-assembly. This is an example of the supermolecule, and sometimes it's called self-assembly systems. And these are the papers, if you are interested in, we will discuss those in these papers. Those will be the papers discussing the structure of the supermolecule. And if you are interested in the mathematical part, this is a paper. You need to take a look at it. Okay, so the supermolecule is made in Fujita Lab in University of Tokyo. And they will use metal ion and also organic ligand and to make a complex that is called supermolecule. And most of the time, they make very complex polyhedral structure as in this picture. But today, we are interested in a supermolecule which have the link structure. So these are the examples of the supermolecule which have the link structure. The first, there are 2 papers, and the first paper will consider supermolecule made of 4 components. So these are the links made of 4 rings, and we will discuss the difference of the topology of 2 complexes. And the second one is this one. So this is very complicated, but this is made of 6 rings, And we like to discuss how this is made. Okay, the first one is this picture. So this is made of Ag plus metal ion and also peptide, tripeptide as in this picture. So P is L-fluorine and the G is glycine. And if you prepare PGP tripeptide with a pyridine here, then this pyridine part will connect to Ag plus, and that will make a form as in this picture. So this is a, if you prepare, so this is a PGP, and this is a Ag plus ion, and the three of those will make one ring. So if you prepare how many? 12 of the peptide and also 12 Ag plus, then that will form a 4-component ring. And this is the one supermolecule they make. If you prepare this one. So if you prepare a PGP, this is the topology they have. And you can change the peptide. If you take threonine instead and the TPP, then the topology will be changed. And this is the picture. So the start of the collaboration of the work, they asked me these questions. So they make very interesting supermolecule, so this is a topology of the supermolecule. They think that those topology looks like different, but they don't know how to show those are actually different. So when you consider in a topology of the molecule, you can maybe it looks different, but maybe you can just move this one into this one. So actually, if you like to say that those topology are different, you need some proof to show those two topology are different. And the question is, so there are two questions. That is this one. And also, the first question is the complexity of this, the supermolecule. If you draw a diagram of this link, so this is a diagram, it seems that there are 12 crossings. And the question is that if you consider a minimum crossing number, that is crossings among all the diagram of this link is actually 12 or not. So those are the questions they ask. And we like to answer this using knot theory. For the first question, determination of the crossing number, we can use a result on the alternating link, and we can give the affirmative answer to the question. The observation is that the following. If you draw a diagram, as in this picture, we can see that those are the alternating diagram. The diagram is alternating if you move along each component link, then at the crossing, overpass and the underpass comes in order. So over and under. Then that diagram is called alternating. And in knot theory, many things are known for alternating knots and links. For example, if you consider crossing number of the alternating knots and links, then by a result by Kaufman, Lasky, and Scissorswaite, 1987, it is easy to check the minimum crossing number for alternating knots and links. So if you have a reduced alternating link diagram, then that attains the minimal crossing number. The alternating diagram is called reduced if there is no Nugatori crossing are in this picture. If you have this crossing, then you can just twist and reduce the crossing number. And those diagram is called non-reduced. And if there is no such a crossing, then it is a reduced alternating diagram. So if you take a look at this picture, then those two are alternating diagram without Nugatori crossing. So these are the reduced alternating diagrams. Then from this picture, it is easy to see that the crossing number of these links are actually 12. So those two links have the same crossing number, 12. So that will answer the first question. And we'd like to give the answer for the second question. The topology is different or the same. To do that, we observed that the topology of this link, and we found that actually these links can be made from using a polyhedra, and those links are called polyhedral links. In an experiment of the supermolecule in a Fujita lab, they often make polyhedra, and they know the crystal structure of those supermolecules, and they know the symmetry group of the supermolecule. And in this example, they know the symmetry of these links. They found that they have the tetrahedral symmetry. And we like to give the background why the tetrahedral symmetry comes from. And we found that these are made from polyhedra. So in this case, using tetrahedron, we can construct those two links. So I'd like to introduce what a polyhedral link is. And so I will go back this later. Maybe I should discuss this. I think I should go to the definition of the polyhedral links and go back to this one. Polyhedral links is made in this way. First, you prepare a polyhedra. So this is just a part of the polyhedra. So if you consider the tetrahedra or the dodecahedron or ixohedron, then you have some face. So in this case, you have triangle. And this is some edge. And at each edge and crossing, you change the part as in this picture. So in a type A polyhedral link, for each edge, you put two crossings. So here, this edge will change into two crossing. And so here, edge, the vertex is changed into this one. And if you change this, then you have this structure. On type B, the edge will change into just a two line, and at the vertex, you put three crossings. Then you have this structure. This is called type B. And for type C, we just consider some special polyhedra. Each vertex, you have degree 4. That means that there are 4 edges connected to a vertex. Then you can do this type of the change, and then you have some link that is called type C polyhedral link. And we found that those supermolecule is actually made of this type A and type B. And A will give planar structure and type B will give pyramid. So here we just draw a picture along just a face. So if you consider a face, along the face, you put this. But if you consider the whole polyhedron, then the structure will be curved, and you make a very closed form. And okay, go back this. the first one is made from tetrahedron, and if you do type A, then you have two crossing at the edge, and no crossing at the vertex. Then you have this picture, and you have the supermolecule of this one. And if you do the type B, then you have this picture. If you consider whole tetrahedra, and you have this picture. So we found that these are made of polyhedral rings. Okay. And the third one is very complicated, but you prepare a cube, and if you do the type A, then you have this picture. So this is made of cube, and this one has very interesting property. So this is has a capsule structure, and you have a hole inside. This is the size of the supermolecule. Inside you have a hole of diameter 1.6 nanometer, and the diameter of the whole structure is 3.7 nanometer. Okay, so this is a example of the using knot theory to explain the topology of the supermolecule. And using the knot theory, actually we can say that the topology are actually different. And if you prepare a very complex polyhedron, as in this picture, this is a truncated octahedral ring, then you have this picture. So this diagram is flattened, but if you make it in a real way, then you have some capsule structure. But we often use this diagram to study knot theory. And if you make a polyhedron, then we can have the information of the crossing number and the number of the links, as in this picture. So this type of links is studied in a Chinese group in 2009. And if you make a type A polyhedral link, then the number of the components, that is the number of the links, is the number of the face of the polyhedron. And also the crossing number is 2 times of the number of the edges. So if you know the construction, we know all the information of the number of the rings and also the crossing number. And actually there are many polyhedrons. So in this picture, example of the regular polyhedra, there are 5 of those. And also, so from this, you can make this type of polyhedron. And this orange one is actually made in a Fujita lab. So from the tetrahedra, they make type A and type B. Those are the first 2 examples. And from the cube, they make this one. And these are the second one, very complicated. And if you prepare those dodecahedron or ixohedron, you can also make a different knots and links, different links. Okay, so this is the example. And actually, you can prepare a different one, semi-regular polyhedra. Then you can also make a different knots and links. And these are the example. If you make a polyhedron, then these are the information. If you use truncated tetrahedron, then if you make a type A, this is the number of the rings and the number of the crossings. So if you prepare a very complicated polyhedra, then you have a chance to make a very complicated supermolecule. And sometimes if you prepare a different polyhedra, but you will have the same topology. So if you prepare a cube and the octahedron, and if you do the type B construction of the polyhedral link, then it turned out that those are the same. Sometimes you have the same topology even if you prepare a different polyhedron, but most of the case we can show that the topology of the polyhedral links are different. So those are actually the very special case. And what we can show mathematically is the following. If you prepare some polyhedron, so like a tetrahedron, and if you make a link type A and type B from this tetrahedron, then we can show that those two links are different. Those are equivalent links, but they share the same property. So in this case, the number of the crossings is the same. So this will give you the answer of the first question. These two links are different. And if you prepare polyhedron with degree C, then we will have the two different links with the same number of the components and the same number of the links. So this is the answer for the first question. And using this, you can make some supermolecule which share the many property, share the many same property, but the topology are different. Okay, I'd like to give the idea how you prove this. So this is a mathematical method we will use to show those actually two links are different. So when you have alternating links, we know classification of the alternating links. That is called affirmative answer of the Tate-Flapping conjecture. Suppose you have two alternating diagrams of the same link. Then you can change one diagram into the other just using this type of the operation. This is a flip operation. So here you have some part of the diagram, and you flip this part in 180 degrees, then this part is changed into this part, and also this crossing is moved to this crossing. And this is called flip operation. And if you have the same link, the two diagrams the same link, then you can change one to the other using only this. And we show that if you consider type, so we will show that if you have alternative diagram of type A and type B, we can show that there is no such flips. So for that, we will use dual graph to check where the flip is. But we don't go into the detail. But this is how we show those two links are different. Okay, so this is the one example of the application of knot theory to supermolecule. We can show that actually two topology are different, and also we can give some idea to make a new molecule. The second one is application of the knot theory to DNA and vortex. And these are the two papers we will discuss. First one is 2013, and second one is 2017. So here we consider the topological change of the DNA. So here we consider double-stranded DNA, but so in this talk, we will consider a closed circular DNA. So DNA is closed, and we will consider the topology of the closed circular DNA. And most of the case, we will just draw a picture which indicates the center line of the double-stranded DNA. So in this case, the yellow line. And this is a picture of the knotted or linked DNA. So this is taken in 1983. This DNA is protein-coated, actually, regular protein-coated DNA. So DNA is fragile, so you need to put some protein to take a picture. So, but you can see some topology of the DNA. And actually, DNA is knotted. And so this is an idea by Ernst Sumner's. If you consider enzymatic action of DNA, so in this case, we will consider site-specific recombination. That I will explain later. If some enzyme that is called recombinase will act on the DNA, sometimes it changes the topology of the DNA. So first, you have the substrate. This is just a trivial knot. The topology of this DNA is just a trivial knot. After the recombination, as in this picture, you have a different topology. And Ernst Sumner used the information of this topology, the topology of the substrate, which is DNA before the recombination, and the topology of the product, which is DNA after the recombination. They will give the information of how the recombination acts on the DNA. So we like to draw a picture what is happening inside this black box. This is the part where the enzyme acts. So this is a brief introduction of the site-specific recombination of DNA. Enzyme will grab some part of the DNA. So here you see just a part of the double-stranded DNA here and there. The enzyme grabs two parts of double-stranded DNA here and do the recombination as in this picture. As a result, this part is connected to this part, and this part is connected to this part. And this will change the sequence of the DNA. And site-specific means that the enzyme actually recognizes the sequence of the DNA that is called site, and the enzyme acts only on that specific site. And that recombination is called site-specific recombination. This is another example of the site-specific recombination. The enzyme grabs this part and cut and turn 180 degrees and reconnect. Sometimes this will happen. And this is a very schematic picture of the site-specific recombination. So this is a site, and this is a double-stranded DNA. The enzyme grabs this part and cut here and turn 100 degrees of the top part. And as a result, you have this picture. Then the sequence of this part is inversed. So you have the different sequence of the DNA. And if you see this picture, the site has the orientation, and orientation is very important. And in this case, this is direct repeat, and from this, after this recombination, the number of the DNA doesn't change, but if the site is placed in this way, this is called a direct repeat, then as a result, the number of the component of the DNA will be changed. So in this case, this part is cut, and you have two components of DNA. Okay, and we will use mathematics to model this DNA recombination. And here we will use something called a band surgery. And this is a DNA before the recombination. And this ball graph indicates the black box part. So here the enzyme acts. actually, the enzymatic complex grabs two strands of the DNA and changes, as in this picture. They cut this P part and reconnect in this reconnect as in this way, and you have R. And by this operation, sometimes the topology of the DNA will be changed. And we will use the information of this topology, and we like to show what is happening inside this ball. So this is an example of the answer using mathematics. So in this example, the first topology is a trivial knot. And as a result, you have a link, two-component link, and that is called torus link to 4. And using mathematics, we will show the black box part is here. The enzymatic part grabs part of the DNA. Sometimes it also grabs some crossings, and these crossings actually gives will change will be the reason for the change of the DNA topology of the DNA. So this is a schematic. This is a molecule. This is a picture of the inside the enzymatic complex. So yellow one and the purple one is a double-stranded DNA, and the green one is an enzyme which acts on the DNA. And these are protein which will be inside the enzymatic complex that will help the enzyme act. And so this part, the enzyme acts and cut the DNA and do the reconnection as in this picture. And we like to draw this kind of picture as a result of the problem. And also the similar things, things also happen to the vortex knot and links. So if you prepare a vortex knot, then by the reconnection, the topology of the vortex will be changed. So this is a picture from the paper in 2013. So they prepare torus torus knot as a vortex knot, and they observe that the topology will be changed, and you have a hop link and a trivial knot, and also a trivial link. And they also have a video of the experiment. So first, you need to make this very nice tool to make very complicated vortex. So here you see a vortex of the triple knot. And the reconnection happens afterwards. Reconnection happens here, and the topology of the vortex will be changed. And actually, there are several reconnections. In this case, maybe 3 of those. And you have a link, maybe trivial 3-component link afterwards. So you can see that these are also very similar what is happening for the DNA recombination. So in this case, this topology change is called reconnection. And in their experiment, first they prepare a triple knot. And the first reconnection will change into a hop link and a trivial knot and a 2-component trivial link. And we can also use a band surgery to model this. I will explain the band surgery later. So this is an experiment in 2013, and there is a simulation, a recent one, 2012. They also, this is from Lorenzo Ricca's lab. They prepare a trivial link first, but you need to put it in a very good way. And the simulation is for the quantum knot. And for this, first you have trivial link. And then after the reconnection, you have a trivial knot and hop link and trifold knot. Sometimes the complexity of the knot and links goes up. So this is observed by simulation recently. So we like to model these phenomena using band surgery. So band surgery is defined as in this picture. This is a part of the knot or link. You put a band as in this picture. This is just a square, but is a half-twisted square. And two sides of the square is connected to some part of the link. So in this case, this side is connected to this side here, and the other side is connected to there. And for the square, two sides is connected. And what we do is that we remove the two sides, and instead we put the other two sides to the knot or link. Then you have this picture. This part is cut, and this part is connected. And that will change the topology of the knots and links. And this operation is called band surgery. Using band surgery, you can change the knots and topology of knots and links. So this is an example. And we are interested in coherent band surgery. In many cases, physical knot have orientation. For example, if you consider DNA knot and links from the sequence, you can put the orientation of the DNA. And if you consider vortex knot and links, then that has natural orientation where the vortex moves. And using that orientation, we can put orientation of knots and links. And the coherent band surgery is defined as in this picture. That will preserve the orientation outside the part of the band surgery. So here, the orientation, suppose the orientation comes this way and also this way. And afterwards, you have different link, but this also has an orientation. And the orientation is this way and this way. Then this band surgery is called coherent. That will preserve the orientation outside the band surgery. So this is an example of the coherent band surgery. So this is oriented oriented torus knot. And if you do the band surgery here, then you have this picture. This is an oriented hop link. Then this band surgery is called coherent band surgery. Okay, the first observation is that if you do the coherent band surgery, it always changes the number of the component. If you start from torus link to 6, as in this picture, and if you do the coherent band surgery, this is an example of the coherent band surgery. If you do the coherent band surgery here, then you have this picture, and then this crossing will be erased. Then you have this. Then from the two-component link, you have a knot, and so on. And if you consider DNA, this is two DNA which is linked. Then you have a dimer, just a one-component DNA, and so on. Okay, using this band surgery, we like to characterize a unlinking pathway of DNA catenon. This explains where the DNA link appears. If you consider a closed-circular DNA and in a cell, and if you consider the cell division, this DNA will be replicated, and that happens. This is a picture of the DNA replication. The double-stranded DNA will be cut in half, and the copy will be made. And if you like to replicate this closed-circular DNA, all you have to do is just cut the double-stranded DNA along the yellow line. Then, because of the topology of the double-stranded DNA, you have you can show that two new DNA will be linked as in this picture. But in a cell division, if there are two new DNA will be linked, that is a problem. Each component of the DNA will go to two different daughter cells. But if it is linked, you cannot do it. So that will be a very big problem. And in a cell, enzyme which is called type 2 topoisomerase will unlink this type of linking. And this enzyme will do the crossing change as in this picture, and that will unlink this two-component link. But in the case the topoisomerase is not active in a cell, a cell tries to survive in a different way. And it is observed that if there is no topoisomerase, then different enzymatic action that is called XCL FTSK recombination also unlink the linked DNA. So they show that if you prepare this link, then this enzyme also unlink the DNA. And so this is the experiment. They prepare some links, and after recombination, you have a trivial link. The mathematical problem they answer us is that they don't know how it is happening, and they know the first topology and the last topology, and we like to know what is happening between them. So we need to characterize using knot theory. And from the experiment, we give some mathematical assumption. From the experiment, we give assumption that at each recombination, it seems that the complexity of the linking that can be observed by a crossing number will go down in each step. And using this as an assumption, we will characterize unlinking pathway. So this is the example of the unlinking pathway. Starting from torus link to 6, by one recombination, you have 5 crossing. If you do a band surgery here, then you have this, and you have dimer. And the second step, if you do the recombination here, then you have this. And in this case, you have 6 steps to make it unlink. So this is one example. And it seems that this phenomena will happen in a cell, and we have to give this will be the answer using mathematics. Okay. So this is the problem we like to consider. The first, the link is given. So in this case, we consider 2,6 torus link. And you do the band surgery, and we assume that as a result, we have a link, we have a knot, one component knot. And we assume that the knot has at most 5 crossing. The complexity goes down. Then only using this, we can give an answer. And we can show that the result will be torus knot 2,5. So this is the answer. So this is one example of the application of knot theory to this. And in the next step, you have this knot. And also suppose that the crossing number goes down. So you have at most 4 crossing. And in this case, you have 2-component link. Then we can give the answer. As in this picture, the product must be 2,4 torus link. And this has a parallel orientation. And actually, we can prove this in general. Suppose you have 2n torus link. That means you have 2n crossings. Then, as a result, we can show that this is so in this case, the crossing number, sorry, this is not correct. The crossing number goes down by 1. And if you start from this, then also the crossing number goes down. And as a result, we can show that starting from this to go to a trivial link, then this should be the answer. And actually, this is consistent with biological experiment. And if you, so maybe this is a very beautiful, but the assumption might be strong. And in the next step, we just relax some assumption that is, the closing number goes down or stay the same. Then we can also characterize the pathway. But in this case, the starting point, we must restrict ourselves to the topology of the 2,6 torus link. Then we can show that there are 9 shortest pathway. So we only consider shortest. That means this will be unlinked in 6 steps. Then we can show that there are 9 pathways. If you consider all the possibility, actually there are more than thousands pathway from here to there, even if you consider this assumption. But we can show that only 9 will be possible. I think it's time, and I should stop here. But for the proof of this theorem, we will use something called signature. Signature of the knot. That will be defined using Zyphus service. And if you are interested in, you can find the definition in a usual textbook of knot theory. Okay. So and also you can use Jones polynomial, showing there is no such a band surgery from one link to the other. Okay, I think it's time, and I should stop here.
Thank you so much. Let's thank our speaker for this wonderful lecture.